So our first speaker is Professor Gulin Oz from the University of Minnesota, who is going to talk to us about non-invasive imaging-based methods to measure glycogen and related phenomena.
I'm very new to the APBD area, but I know something about glycogen. I have studied glycogen using MR spectroscopy, and Jesse asked me to give you a little bit of background on MR spectroscopy and also different ways to use the techniques to perhaps address the questions in APBD and maybe pursue some biomarker. Quick background on MR spectroscopy, and I'm starting out with the most commonly widely used MRS modality, and that's Proton MR Spectroscopy. So essentially you use the same MR scanners that are used in the clinic. Here I have a picture of a 3 Tesla scanner, but you use it to obtain information on neurochemicals, neurochemistry in the brain rather than images. So in this example, I have a structural image that everyone is familiar with, and then you select a region of interest, like shown here, and then acquire a spectrum. This is a 4 Tesla spectrum, proton spectrum that has signatures of a list of neurochemicals that are concentrated enough to give an MR signal. So each of these peaks comes from a different neurochemical. For example, this one is N-acetyl aspartate, the most widely validated neurochemical to be a marker of neuronal health. But then we have other neurochemicals like creatine, phosphocreatine, choline, etc. We have neurotransmitters that we can access and quantify, such as glutamate and GABA. We have some antioxidants, glutathione, vitamin C, etc. And the intensity of each of these peaks is proportional to the concentration of the neurochemical in this region of interest. So you do a fitting procedure, and then relative to water that's acquired from the same volume of interest, you quantify, you get millimolar quantification for this list of neurochemicals. So just to give you an idea on what cellular and biochemical processes are measurable by high-field proton MRS, changing in cell number and viability. For example, we have some neuronal markers, N-acetyl aspartate that I already mentioned, and glutamate. Most of the excitatory neurons we have in the brain are glutamatergic neurons. So if you have any neurodegenerative condition, NAA and glutamate tend to go down together. Then we have putative glial markers, myonostal and glutamine, that tend to go up if there is gliotic activity in the brain. We have potential markers of demyelination and increased membrane turnover. Those are choline-containing compounds and mobile lipids that become detectable in certain conditions. Defects in energy metabolism can be indicated by changes in creatine, phosphocreatine, lactate, and glucose. And oxidative stress markers, the 2 highest concentration antioxidants, glutathione and vitamin C, are detectable by this method. So here's an example from another set of diseases through which Jesse and I met, spinoservular ataxias. And I have on the top here spectra obtained from the cerebellum in one control individual and in an individual with spinocerebellar ataxia type 1. This disease results in cerebellar atrophy and dysfunction, which results in ataxia. So even in a single spectrum, you can see the NAA signal is down in the individual with SCA1, as well as the glutamate signal is down. So what I had mentioned earlier about assessing neuronal viability through these markers is apparent here. And in this disease, we happen to see changes both in glutamate and glutamine. And if you look at their ratio, the glutamate-glutamine ratio, you get an indication of neuroglial deficits. So perhaps an abnormal glutamatergic activity can be indicated by this reduced glutamate-glutamine ratio. But then to be more relevant to APBD, the first question that Jesse asked me was, okay, is glycogen actually detectable in the brain by MR spectroscopy? And the answer is yes. We already saw in one prior talk, I think that was a muscle rodent application. And this is doable in humans too, but you need to label the glycogen molecule with carbon-13 labeled glucose. So, I should say these experiments really are not widely available. Our team in Minnesota has so far been the only team who has done this kind of work in human subjects to look at glycogen and glycogen turnover using MR spectroscopy. So this is from our 4 Tesla work. Using a surface coil, we are looking at the back of the brain here, and we are detecting the signal from this very large volume of interest right here. So it's a good portion of the occipital lobe because the signal is very weak. And we can do this because glucose, when stored in glycogen, gives rise to a separate peak right here. This is a C13 spectrum. So these two are coming from the free glucose, and this peak comes from glycogen that's labeled with carbon-13 labeled glucose. So, and the problem with brain glycogen is, the turnover of glycogen in the brain is very slow. It takes days to turn over the entire pool, and we did not know this. When we started these studies many years ago, early 2000s, when we first wanted to detect glycogen in the human brain, we would administer C13 labeled glucose IV over a couple of hours and wait for the signal. There was no signal. So then we realized, okay, turnover is really slow. It's not like muscle or liver. And also the amount of glycogen that's stored in the brain is much lower than muscle and liver. So we had to design these multi-day long studies where we would infuse C13 glucose, in this case, over an entire day, watch the glycogen signal go up, this peak here, you can see it going up, and then quantify that, and then monitor the washout so that through metabolic modeling, we can get an idea of content and turnover rates. And this is what these data show. So we have 2 to 3 participants in each of these groups. Here, people who have received 6 hours of C13 glucose infusion, 11 hours, etc. So, and by combined modeling of these data by looking at label incorporation and then washout, and this kind of pretty simple metabolic modeling, we estimated glycogen concentrations. This is in terms of glucose units stored in the form of glycogen in the brain to be about 4 millimolar. And then we got a time constant for turnover that was almost a day. That means to turn over the entire pool would take 3 to 5 days. So it is a significant glucose reservoir relative to free glucose in the brain, which is 1 to 2 micromoles per gram, but it turns over very slowly. So before I go there, so this technique is available, but in the biomarker setting, I don't think this is applicable because it does take a whole clinical team to administer these the C13 labeled glucose, and it takes several days to get the full information. So then Jesse was wondering if there are other methods to look at energy failure, to assess metabolism through MRS in the brain. And yes, you can use C13MRS to look at oxidative glucose metabolism as well as glutamate-glutamine cycling rate. And the idea is the same. You administer C13 labeled glucose IV and then watch the label go essentially through TCA cycle. TCA cycle intermediates are too low concentration to be detectable by MRS, but then once the label reaches glutamate down here and then gets exchanged with glutamine, those are the metabolites that you can detect. So metabolites that are in exchange with the TCA cycle. And aspartate is another one. So those three are detectable. And again, through metabolic modeling, one can measure cerebral metabolic rate of glucose and also glutamate-glutamine cycling rate. So this is a two-compartment metabolic model. You have the neuronal and glial compartments. People have applied this method in clinical cohorts, not through extensive modeling, but just by looking at label incorporation over time and detecting differences in this particular case between a control participant and an individual with Alzheimer's disease. If you look at just the glutamate signal, for example, right here. Let's see, okay, there's my mouse. So look at the intensity of this peak relative to everything else. In the control participant, it's much higher. I mean, there's more metabolism, there's more label incorporation into glutamate. So that's one method. Still, it takes a whole team for the IV administration of C13 labeled glucose to participants, but it's one scanning session, 2 to 3 hours, and then you have your information. Then a little bit simpler method to get an evaluation of energy status in the brain, it's PhosphorusMRS. So in this case, you are detecting endogenous signal. You do not need to provide a labeled compound, administer labeled compound, but it requires non-standard hardware. So you can detect ATP, ADP, phosphocreatine levels, and here's one example where they detected differences between controls versus patients with Parkinson's disease. And this is a spectrum that my colleagues acquired at 7 Tesla using PhosphorusMRS. Now, in general, you don't expect changes in ATP, ADP levels in the brain because the homeostatic pressures do keep them at normal levels until serious energy failure. So these results that came out in Parkinson's disease have surprised me, but they seem to be valid and they have been replicated. So this is another modality that can be applied to look at energy status. And then finally, again, thinking about having a handle on energy metabolism, it is possible to measure cerebral metabolic rate of oxygen, this is oxygen utilization rate by O17 MR spectroscopy. And what you're doing in that case is using O17 gas through inhalation and monitor the flow of label into metabolic water. And this is so far primarily available in animal models. It's not a widely available technique, but I have colleagues at our center who routinely do this. They have shown the feasibility in humans, but oxygen 17 is very expensive. So for as a biomarker in humans is probably not very appropriate. So yeah, the idea is oxygen O17 gas is NMR invisible, once it's incorporated into metabolic water, it's NMR visible. And this is how the data look like. These are dynamic experiments. This is the baseline. There is very little amount of O17 natural abundance in water. So this is what you're seeing baseline, and then the animal, in this case a mouse, inhaling over 2 minutes. This is very quick measurement, over 2 minutes, oxygen 17, and you reach a new steady state. And from this slope, you can calculate CMRO2. And this is done in chemical shift imaging form anyway. So you get maps of CMRO2 in the mouse brain.